Welcome to section 3 of reproductive embryology. In this section, we'll be discussing embryologic derivatives. Let's get started. Here is an overview of what we'll be discussing in this lecture. Step 1 expects you to be able to differentiate between embryologic derivatives in questions by identifying the origin of certain adult structures. The three layers of the embryo consist of the endoderm, which is the internal layer, mesoderm, which is the middle layer, and ectoderm, which is the external layer, and includes the structures of the CNS. In this video, we'll start with the innermost layer, the endoderm, and work our way to the external layer of the embryo, which is the ectoderm. To understand the three body layers, it's important to understand how they are established in the first place. We covered this image in section 1 of reproductive embryology, but I'd like to focus on this part right here. Recall that the formation of the trilaminar disc happens through a process called gastrulation. Gastrulation occurs during the third week of development and is the process of taking the bilaminar embryonic disc and forming a trilaminar embryonic disc. This process is made possible by a longitudinal midline structure called the primitive streak, which we talked about in section 1. So as the cells from the epiblast migrate through the primitive streak, which we can see right here, they form the endoderm, mesoderm, and ectoderm, which we can see right here. Okay, now that we understand how the body layers are established through the process of gastrulation, let's talk about the adult derivatives of endoderm. A mnemonic that some students find helpful is internal layer. In other words, the endoderm forms many of the body's internal structures. The main structure that the endoderm forms is the gut tube, which we discuss more in our video on gastrointestinal embryology. This includes structures such as the gut tube epithelium, liver, gallbladder and pancreas, as well as other GU structures like the urethra and lower vagina. Additionally, the endoderm forms the lungs, including the alveolar cells, the cells associated with the thyroid and parathyroid glands, and finally the eustachian tubes and the thymus. Think of the mesoderm as the middle layer of the body. This includes muscle, bone, and connective tissue, including the dermal layer of the skin, the serous lining of cavities, including the peritoneum, pericardium, and pleura, the spleen, and this is an important structure to differentiate as mesoderm rather than endoderm because many of the other GI structures are derived from endoderm. Other structures include components of the cardiovascular system, including cardiac muscle cells, blood and lymphatics, the wall of the gut tube, genitourinary structures, including the upper vagina, testes, and ovaries, and finally, the kidneys and with them, the adrenal cortex. This is a long list of structures to remember, but if you take away one thing, I would remember the muscle, bone, and connective tissue, as well as the spleen. A popular mnemonic to remember mesodermal defects is VACTERL, which stands for vertebral anomalies, anal atresia, cardiac defects, TE fistula, renal defects, and limb defects. This group of defects is considered an association, which just means that it's a group of birth defects that co-occur with no pathologic explanation. In this case, the association between each defect is the fact that they are derived from mesoderm, not because of a specific disease. This is an image of a newborn with limb anomalies due to a mesodermal defect. Remember the L in VACTERL stands for limb anomalies, and in this case, the patient has a limb anomaly due to a mesodermal defect. Okay, moving on, the ectoderm is composed of three components: the surface ectoderm, neural tube, and neural crest. Let's talk about the surface ectoderm first. The surface ectoderm can be considered the external or outer layer of the body. It includes the epidermis. The anterior pituitary arises from Rathke's pouch, and this pouch is an invagination at the roof of the mouth in front of the buccopharyngeal membrane. It gives rise to the anterior pituitary and technically part of the endocrine system. A notable malformation of Rathke's pouch is a condition called craniopharyngioma, which is a benign Rathke pouch tumor with cholesterol crystals and calcifications. We talk more about this in endocrine embryology. Surface ectoderm also gives rise to sensory organs including the lens of the eye, the epithelial lining of the oral cavity, and the anal canal below the pectinate line. Finally, glands including the parotid, sweat, and mammary glands are important contributions from the surface ectoderm. Now that we've covered surface ectoderm, let's discuss the next part of the ectoderm, the neural tube. We discussed the neural tube, including malformations in more depth in the video on neurologic development, but I'll give a brief overview here. The neural tube is an incredibly important structure in development as it gives rise to the brain, retina, and spinal cord. An important association to make is that the neural tube is part of the neuroectoderm, which contributes to the structures of the central nervous system. Failure of the neural tube to fuse results in neural tube defects, such as spina bifida, meningocele, meningomyelocele, and anencephaly. Folic acid is important in the proper development and closure of the neural tube, and many of these malformations are a direct result of folic acid deficiency. This diagram shows us where the neural crest cells and neural tube come from. During neurulation, the borders of the neural plate, also known as the neural folds, converge in the center, creating what's known as the neural groove. This convergence continues until the neural groove eventually fuses and forms the neural tube. After gastrulation, we're left with the surface ectoderm, the neural tube, and neural crest cells. The neural tube and neural crest cells together are referred to as neuroectoderm. As you can see, neural crest cells are located at the neural plate border right here. The neural crest cells delaminate and migrate towards the periphery, eventually giving rise to many cell types. Here we can see a list of the structures derived from neural crest cells. They make up a diverse amount of cell types, and they can be remembered with the mnemonic motel passes. This includes melanocytes, odontoblasts, tracheal lining, enterochromaffin cells, leptomeninges, PNS ganglia, adrenal medulla, Schwann cells, the spiral membrane, endocardial cushions, and skull bones. In reality, memorizing all of these is probably not the best use of your time. For examination purposes, you're usually only tested on a few of these. In my experience, the PNS ganglia, spiral membrane, and endocardial cushions are the most important to remember. The PNS ganglia should make you think of the neural crest cells that migrate to the colon, and when defective can result in Hirschsprung's disease. The spiral membrane and endocardial cushions are embryonic heart structures, and when defective can result in congenital cardiac diseases, such as tetralogy of Fallot and septal defects. I discuss these in much more detail in the cardiac embryology lecture. So if I were in your shoes, I would spend my time focusing on these, as these are probably the most clinically relevant. All right, now that we've covered the material, let's review with a question. A 7-year-old boy is brought to the physician due to shortness of breath and chest pain. Upon further investigation, he is given a diagnosis of sickle cell disease. What is the embryologic derivative of the cells affected by individuals with this condition? Okay, from the question stem, hopefully you notice that this boy has sickle cell disease. In this disorder, the red blood cells are affected, and recall that red blood cells are derived from mesoderm. So the answer to the question is mesoderm. And with that, we've covered everything you need to know about embryologic derivatives.